Hi everyone, this is Andy from Med School EU, and in today's video we are going to discuss the genetic basis of evolution. Be prepared, there's going to be plenty of definitions for this topic, and I would say get a pen and paper so you can take some careful notes. So first, we are going to discuss some notions of evolutionary process. So there's a couple of things that we need to define to understand what they are, what they mean, and what they entail. So first, we're going to define natural selection. And natural selection really increases the frequency of certain alleles. So increases, that is called natural selection. In the last topic, we should have discussed artificial selection, but I left it out to talk about it in this video specifically because we are talking about natural selection as we're defining it. And artificial selection will be aimed at the same thing, that increases the frequency of certain alleles. However, it's not done naturally by the environment. It is done artificially in a laboratory or by other means of reproduction. For example, a lot of our crops and a lot of our products that we eat are artificially made. They would have the GMO label on them, genetically modified organisms. So that would be a way of, that would be an example of artificial selection. And sometimes on the exams, there are questions that come up about natural versus artificial selection, and they're quite easy to distinguish. So I'm not going to go into too far detail about that. However, another notion about natural selection is these certain alleles they're just the alleles that contribute to reproductive success in a particular environment. So alleles that increase fitness. So that is the definition of natural selection. It's pretty self-explanatory. It's just that the environmental pressure causes certain offspring or certain individuals to die and certain individuals to survive and reproduce. And the ones that will survive and reproduce, they will obviously increase the frequency of their alleles in the population because those are alleles that will be passed on to the offspring, and then therefore those offspring will also be able to survive the natural environment because they have the alleles of the parents that did survive the environment and were able to reproduce. Now looking at the next definition, and that's going to be genetic drift. And this one is a little bit different than natural selection because it causes a change in alleles or an allele frequency that is completely random. So in some cases, drift or the genetic drift may cause alleles that decrease fitness to increase in frequency, or it could be completely the opposite. And we're going to talk about a couple of mechanisms that occur with genetic drift. And the third definition is called gene flow. And this occurs when individuals leave one population, join another, and breed. So if you have two different populations, for example, there are wolves in North America and there are wolves in Europe, for example, and if you take some wolves from Europe and you put them on a ship and bring them to North America, and then they are going to be integrated into the wolf population here in North America, and they're going to interbreed, that is called gene flow because they're bringing the genes from Europe over to North America. And that's really the basis of gene flow because those wolves that are coming from Europe will introduce new alleles to the North American wolf population. So there are two things that cause genetic drift that we must discuss to really understand what genetic drift is. Two major things. One is called founder effect. Founder effect. And the second one is called genetic bottleneck. And this is going to really shine a light on how genetic drift really works. How can it have alleles, increase the frequency of alleles that are bad for the fitness of the organism. The founder effect is basically the same as the notion that we talked about gene flow, because it is when there's a change in allele frequencies that occurs when a new population is established. So with founder effect, if you have immigration, for example, from one population into another geographical location, right? So if you have here, we have a population of plenty of different wolves in North America, right? And now all of these wolves are going to go over to or some of these wolves are going to go over to a small island, and they're going to interbreed. Now this part is going to have new alleles, because allele frequencies are going to change because there's less variation going on, because some of them immigrated over to a new geographical location. That is the founder effect. Now, if we're talking about genetic bottleneck, we would have the same environment, so the same geographical location, except that plenty of these organisms are going to be killed off. Maybe there was some sort of storm, or maybe there was a shortage in food or shortage in water, and a lot of them don't survive. So we take off a big chunk of population and we only have a couple left. That's what occurs with genetic bottleneck. So there's no immigration, there's no migration. It's simply the same geographical location. However, there was some sort of adverse event that occurred and it destroyed a big chunk of the population. So there's less individuals now, or there's less smaller population, and therefore they are going to create new alleles now, or allele frequency, because there's just a smaller population. Next, we're going to discuss the mode of selection. So this is best explained with a diagram here, because there are four types of modes of selection, at least four of the most common ones, and we're going to discuss each one and their significance. So this first one on the left that we have is called stabilizing selection. The effect that it has on the phenotype is that it favors phenotypes near the middle of the range, so near the middle here. That's the ones that it favors, and it just maintains the average phenotype. So as an example, human babies of average size are most likely to survive. The big babies are less likely to survive, and the small babies are less likely to survive compared to the average size. That's going to be called stabilizing selection. And the genetic variation due to this stabilizing selection is reduced, meaning how many different alleles we are going to have. Because of course, the more different types of alleles and combinations of alleles that we have, the higher the reproductive success of an organism. So here, in stabilizing selection, it's going to be reduced. And that's pretty obvious because it's simply going for that one average. It's not focused on any of the other particular alleles. So the other alleles will be killed off and the average will thrive. Now looking at the other selection, the one that's pushing it to the side. So the yellow here is what it was, and now this one is what it has become. And this type of selection is called directional selection. So the effect on the phenotype that directional selection has is that it favors one extreme phenotype, causing the average phenotype in the population to change in one direction. It reduces genetic variation once again because it's simply focused on just the one type of allele that is able to survive, or the one type of frequency of allele. Now, next one is called disruptive And this mode of selection favors extreme phenotypes at both ends of the range of the phenotypic variation. So as you can see here, the average is going to be killed off, but the two extremes will survive. And in this case, the genetic variation is going to increase. And why is it going to increase? Because we are focused on increasing the extreme phenotypes, and we are focused on both sides of the extreme. So if both sides of the extreme are favored, we are going to get all kinds of different frequencies of alleles, which will increase genetic variation. And the final one here is going to be called balancing selection. Balancing selection. As you can see, there is no change, except that it further increases the average selection. It's very similar to stabilizing, except there's a slight difference to it. Is that the effect that it has on the phenotype is basically no effect, because no single phenotype is favored in all the populations of species at all times. So you've got extreme right, extreme left, and middle are all favored. They're all going to thrive. And that's going to be balancing selection, where all babies, small, average, large, are all going to be surviving equally. And this, in this sense, the genetic variation is maintained. So there's going to be no change. It's just going to be the same. The next notion of natural selection and evolution that we're going to discuss is called non-random mating. And there are three different components to it of how non-random mating occurs. And non-random mating is pretty self-explanatory with its definition. So I'm not going to go discuss that in further detail. However, we are going to discuss these three notions of non-random mating. And the first one is going to be inbreeding. And inbreeding is simply mating between relatives. And why is mating between relatives so bad for the survival of the species? Well, and why there are so many defects with babies that have been born from inbreeding. And that is typically because there is no variation in alleles, or there's very little variation in alleles. So if there is inbreeding, the genetics or the gene composition, the allele composition of each parent is going to be very, very similar. And therefore, the baby that will be born will have many chances of developing a birth defect because the gene pool is dramatically reduced. And why does that occur? Well, because there's a decline in average fitness that takes place when homozygosity increases and heterozygosity decreases in a population. So you're not going to get many alleles that are like this, heterozygous. You're only going to get homozygous, recessive, or homozygous dominant. And the elimination of heterozygotes produces many genetic effects. Now, assortative mating is defined as mating that is non-random with respect to specific traits. So this could be phenotypic traits or behavioral traits that would cause certain individuals to choose other individuals that look like them. So again, the law of attraction is that like attracts like. And this is something that would be accompanied with assortative mating, because mates would be looking for other individuals that have specific phenotypes that look very similar to them. Now, that's one way of assortative mating. The other one is completely the opposite, where they look for mates that have behavioral or phenotypic features that would be the opposite of what they are. And sexual selection occurs when individuals within a population differ in their ability to attract mates. And so it favors individuals with heritable traits that enhance their ability to obtain mates. And the next idea that we will discuss is called speciation. That is the production of new species. So first, before we talk about speciation, we should define species. And species is defined as an evolutionary independent population or group of populations. And the key to really defining species is something called reproductive isolation. And this is a very important concept to understand speciation and how it occurs, is by knowing what a species is. And it's basically a population that is reproductively isolated, meaning that there's going to be no other population that can come in and breed with this specific population and produce viable offspring. Because if that was to occur, then they're part of the same species. But if it does not occur and that specific population is isolated from all other populations, then it is its own species. There are two mechanisms for reproductive isolation. One is called prezygotic isolation and the other one is called postzygotic isolation. And these definitions are pretty self-explanatory in a sense that prezygotic isolation is that the two separate populations are not able to reproduce because they cannot form a zygote, and that's called prezygotic isolation. Now, if we're looking at postzygotic isolation, it's in which the hybrid offspring of the matings between those members of different species it does not survive, or it just cannot reproduce. So, for example, the best example would be a mule. A mule is not able to reproduce to make more mules. It is not able to really survive to that age where it's going to reproduce. So a mule is an example of postzygotic isolation because it's not a separate species. A mule is not a species. Why is it not a species? Because it does not have reproductive isolation. It cannot reproduce on its own. The next idea that now we're actually ready to discuss is called speciation, and there's two main parts two main mechanisms for speciation that we will discuss. That's called allopatric speciation and sympatric speciation. And you should know the difference between both because this question actually did occur on one of the IMAT exams, even though this is something that would typically not come up. However, it did, and it's part of the curriculum. So it's important to understand how speciation works and how it occurs with allopatric and sympatric. So speciation that begins with physical isolation, either through dispersal or vicariance, so we're going to discuss those. That's known as allopatric speciation. So this is going to be based on physical isolation. And there's two modes that this physical isolation could occur. And one is called vicariance. So vicariance. And vicariance is just a physical splitting of a habitat. So if you have a specific environment and, for example, a river all of a sudden forms in the middle and the population cannot cross from one side to the other, well, now the two populations are physically isolated. And the other notion is called dispersal. For example, 15 wolves are separated into this little island in the middle of a lake because their path was no longer formed. So, for example, if there's a frozen lake that has an island, a big island in the middle, and plenty of wolves go to this island during the winter. Now, some of them stay there for the winter, and now that the lake is no longer frozen, because it's summertime, they are no longer able to return back to their original environment, so they cannot go back here. Now, you could say that, oh, another winter would come and the lake would freeze again. Well, what if this was just a one-time occurrence and the lake is not going to be frozen to form this path again? It's not going to be frozen for another 40 years until this path is formed again. So they're not able to cross back for 40 years. Now, during that time, it's called physical, they're going to be physically isolated. And now at this point, they may be able to form a new species. Now, it's probably going to take longer than 40 years for a population of wolves to be able to form a new species that would have reproductive isolation. However, if it is occurring for a couple hundred of years, then you might get the notion of speciation in this wolf population, that assuming that it's able to survive, because there's going to be very severe inbreeding going on, especially if there's a small population of wolves. So that's the idea of allopathic speciation. It is all based on physical isolation of a species by the notion of vicariance or dispersal. Vicariance being the same environment being separated physically, and dispersal meaning that one part or a population of a species goes to a completely new environment that is now isolated and they cannot return back. And sympatric speciation is speciation that occurs through populations that live within the same geographical location. However, how does this occur? Well, there's two main events that might trigger the process of sympatric speciation, and that's going to be just external events that disrupt extreme phenotypes, or they would select for extreme phenotypes based on different ecological effects. And the other notion would be internal events such as chromosomal mutations. I'm not going to go too far into detail about this because it is not required. There's quite a bit of research and detail that goes into these ideas of external and internal events in sympatric speciation. The most important thing to realize is that allopathic speciation occurs where there's a physical isolation between a population of a species that may produce new species. And sympatric speciation is speciation that occurs within the same geographical area. There's going to be no change there's going to be no physical isolation. So it could occur through a chromosomal mutation, or it could occur through other external events such as genetic bottleneck. So the last topic for this video that we're going to discuss is called the Endosymbiotic Theory, and that is really the rise of multicellularity that has occurred. And endosymbiotic theory basically states that mitochondria and chloroplasts were originally free-floating prokaryotes. Basically, it is believed the mitochondria are descended from aerobic bacteria, and chloroplast comes from cyanobacteria. So the most important thing to understand about this theory is that mitochondria and chloroplasts did not originally begin within our animal and plant cells. They were free-floating prokaryotes, and then after thousands and even probably millions of years of evolution, it has occurred that they can both coexist under the notion of endosymbiosis by having mitochondria and chloroplasts produce energy, and by having the cell have protection and other nutrients for the original bacterium. Now, another thing that I wanted to mention here, because it does come up, is that mitochondrial DNA is passed on by maternal gamete. And the importance of this is that the maternal gamete, the egg, is much larger than the sperm, paternal sperm or paternal gamete. It's almost twice as big, I believe, or maybe even more than that. And because it's so much larger, most of the contents of the cell, because of course once the two cells fuse, the egg and the sperm, once they fuse, they fertilize, and the zygote that is formed is just going to be a combination of the two cells, right? But the contents of the cytoplasm, so cytoplasmic contents, come from the maternal gamete. And that's simply because the maternal gamete, the egg, is so much larger, and therefore it provides the genetic basis for the cytoplasmic contents. And cytoplasmic contents includes mitochondrial DNA. So the mitochondria that are going to be produced in the zygote are carried by the egg, not the sperm. That's just a general idea. This has nothing to do with endosymbiotic theory. However, I figured I'd point this out since there's no other place for discussion about this particular idea. However, knowing endosymbiotic theory that mitochondria and chloroplasts come from prokaryotes originally, and now they're coexisting within our cells, and knowing their origins, that mitochondria come from aerobic bacteria and chloroplasts come from cyanobacteria, is going to be extremely important for the upcoming IMAT exam. So now that we have completed our unit of inheritance and environment, we're getting to some really exciting topics with human and animal physiology and anatomy. So first, we're going to begin with a video on animal tissues.